So it is coming. Yeah, let's begin. So today's video is about light reaction. So I have mentioned in my previous video, if you're not watched my previous video, I will give you the link in description. It is about photosynthesis. In that I have told you about two types of reactions, and that are light reaction and dark reaction. So in this video we are going to discuss about light reaction. So what is light reaction? What is the main difference? As I've told you in the previous videos, that light reaction occurs in the presence of light, whereas dark reaction occurs independent of light. So today we are going to study light reaction in detail. My handwriting and my drawing is a little bad, so please tolerate for this video, and I think so I will try to improve it later on. So now, without wasting time, let's begin. So what happens? First of all, what is this structure? I want to explain you something. This structure, this wall, this is called thylakoid membrane. Now everybody knows what is thylakoid. I have explained in my previous videos about thylakoid. The thylakoid is a small disc-like structure present in the chloroplast, which helps in light reaction. So in this portion, light reactions takes place. So this is called thylakoid. Now we are going to discuss how light reaction takes place. So it begins from, there are two phytosystems. Let me discuss about the structure first. So there are two phytosystems in this structure. One is photosystem 2 and one is photosystem 1. So the basic thing to note is that the light reaction begins from photosystem 2, and then it proceeds to cytochrome B6 if complex, and then it goes to photosystem 1. So first it occurs in photosystem 2, then in photosystem 1. Other than that, there is a ATP Synthase Complex where ATP is made. So what is the main motive of light reaction? The main motive of light reaction is to produce NADPH and ATP for dark reaction. It means light reaction is a preparatory state for dark reaction. So first of all, what happens? We know the water is absorbed by the roots and is sent to the leaves by xylem. So that water is converted into one water molecule is converted into 2 hydrogen ions and half molecule of oxygen. This occurs in the presence of sunlight, when sunlight reaches photosystem 2. So that at that time it converts into 2 hydrogen ions and half oxygen molecule, and also it produces 2 electrons. Now that 2 hydrogen ions goes down inside the thylakoid, and that 2 electrons flows through the photosystem 1 to PQ. Now what is PQ? PQ is plastoquinone. It is a kind of protein that carries electrons, and it has a capability to carry electrons inside the membrane. It cannot carry electrons inside the thylakoid, but it can carry inside the membrane. So it is called PQ. Then, or plastoquinone. Then later on, this plastoquinone sends that electrons to B6 if complex. In B6 if complex, the electrons are transferred to plastocyanine or PC. What happens then? Why we are transferring it to plastocyanine? This is because, as I have told you, plastoquinone can flow only through the membrane, whereas plastocyanine, it can flow also through the thylakoid, inside the thylakoid. So we need to transport it to plastocyanine so that it travels inside the thylakoid. And plastocyanine is same as plastoquinone, a little difference is there only. And plastocyanine is basically a protein carrier that carries it to photosystem 1. But before that, one more important step is there, that is hydrogen ions that are supplied from outside of thylakoid are absorbed and are taken and are pumped inside the thylakoid with the help of cytochrome B6 if complex. So after that, the electrons move to photosystem 1. Now what happens in photosystem 1? Basically the light reaches photosystem 1, 2 and it reaches every part, but basically it reacts in photosystem 2 and 1. So from photosystem 2 to photosystem 1, the electrons are transported, and then the electrons are transported with the help of plastocyanine. Then after reaching here, they are transferred to ferredoxin with the help of sunlight. Now what happens in ferredoxin? Why they are transported? Because ferredoxin is an iron substance. By the name it's clear, ferri means iron, and then it will lead in easy transport of electrons to NADP reductase. So afterwards, this is transferred to NADP reductase. Now what the NADP reductase do? The NADP reductase, which contains 2 electrons, the 2 electrons which are produced in the beginning are transferred to NADP reductase. Now what happens in the reaction? NAD plus, which is present in the thylakoid or in the leaf, is converted into NADPH when it reacts with 2 electrons and 1 hydrogen ion that is received through this pump or through the first reaction, that hydrogen ion it reacts with and it produces NADPH, which is required for dark reaction. And it is a very essential, NADPH is very essential component of dark reaction. So if we summarize this reaction, it is like this: NAD plus one hydrogen atom produces 2 electrons, which converts into NADPH or nucleated adenine diphosphate hydrogenesis. And NAD plus is NADP. I have not written TP. 1 minute, let me correct it here. It is NADP. So NADP is nucleated adenine diphosphate. So what is the difference between nucleated adenine diphosphate and NAD plus NADPH? The basic difference, as I have told in previous video also, that NADPH is reduced form of NADP, because in NADPH is reduced form as it has an extra hydrogen, so that's why it is a reduced form. Now NADPH is produced. It is one of the most important component, but the other important component is ATP. Now how ATP is produced? That occurs as Atp Synthase Complex. It is also present on the membrane of that thylakoid. So what happens? Basically, some of the hydrogen ions remain, and where they go? They go to Atp Synthase Complex. So what happens in Atp Synthase Complex? There are two main structures. One is F0 or F0, and one is F1 structure. These are only names. So the forward one, which is occurring outside a little, that is F1, and which is occurring inside is called F0. Now basically what happens? The hydrogen ion flows through F0 structure of F0 structure, or I say F0. So F0 structure, and goes to F1 structure. There it reacts with ADP. It doesn't react actually. It helps it to convert ADP and phosphate ion to convert into ATP. As we say, adenosine diphosphate plus one phosphate will form adenosine triphosphate. So it helps in converting ADP and phosphate into ATP, and then that hydrogen is pumped out. So it is kind of transport. It is helping in the reaction. It is acting like a catalyst for the reaction. So with the help of hydrogen only, ADP and phosphate are converted into ATP, and thus adenosine triphosphate and NADPH is prepared. So what is the use of this? These are used in dark reaction, or we can say Calvin cycle or C3 cycle. I have already made a video on that topic. So the next video which I will make will be of C4 cycle. I will not make a video again of C3 cycle. If you want to see that video after this video, I will give you the links in description. So if you like the video, please subscribe my channel and click on the like button. Click on the bell icon if you want to get notifications of my channel. Other than that, if there is any mistake or any suggestion you want to give me, write in the comment section. Thank you for watching the video.